So at this point, we are going to take a look at allergies. Now, allergies are these reactions that develop when a foreign particle come in our body. Now, we talked about immunity before, right? We talked about foreign particles, including bacteria, viruses coming in, and our body mounting a defense against these foreign particles. However, in the case of allergies, the particles we are talking about here are particles that technically are not harmful. However, the body perceives them as harmful particles. They are typically called allergens. So, allergens are going to be the small particles that can come in our system and cause a reaction that we typically call an allergic reaction. Now, allergens are going to be classified based on their size. You're going to have allergens that are large in size, for instance, grass pollen, right? These are going to typically produce hay fever. Then we have allergens that are small in size. This is, for instance, dust mites, cat denders that are actually going to trigger asthma. But what is asthma? Asthma is actually a chronic condition in which allergic reactions are developing all the time, right? And this is going to lead to a contraction of airways, causing a difficulty in breathing. Now, also, we can see, in addition to that, a thickening of the mucus that also is going to have the same effect as the contraction of airways. At the end of the day, it's going to lead to difficulty in breathing. That's why people who typically have asthma are going to take these drugs, which role is to expand somewhat the airways so that the air can flow properly into the lung alveoli. Now, what cells and what factors are involved in allergic reaction? Well, typically, we are going to have B cells that are involved in allergic reactions. In addition to B cells, we are also going to have T cells that are involved. Now, for the non-cellular parts, we're going to have immunoglobulins. And again, immunoglobulins are also called antibodies, right? So immunoglobulins class E, so IgEs, are going to play a very important role in allergic reactions. How are they going to play a role in allergic reactions? By binding on the surface of mast cells. Okay? Mast cells are some type of white blood cells which role is heavily associated with an allergic reaction. How so? By releasing huge amount of histamine and heparin. Now, by releasing a large amount of histamine and heparin, this is going to lead to an inflammatory response, right? Runny nose and runny eye, rashes, and asthma. Now, allergens are going to typically activate helper T cells. Upon the activation of helper T cells, these T cells are going to release cytokines, right? And these cytokines are going to cause a cascade of events, including activations of B cells and other T cells, and not only B cells and T cells, but we're also going to have neutrophils. The monocytes are going to be activated to become macrophages, and so on. Now, there are some allergic reactions that are life-threatening. This allergic reaction that are life-threatening occurs all of the sudden, right? We call this allergic reaction anaphylactic shock. Okay, an anaphylactic shock, it's a very severe allergic reaction that spread throughout the body. They're going to cause an extreme narrowing of airways so that the individual will not be able to breathe in air. And as the exchange of air stops, if nothing is done, the individual can actually die. And that happens more often than we really want to see. Now, there are also some conditions of allergies that are caused by mutations that can occur in some of our genes, including the gene that encode a protein that is called filaggrin. Filaggrin, it's a protein that is typically found in the skin. Now, when the gene that encodes filaggrin is mutated, we can develop a number of allergies, including asthma, but also including allergies to food such as peanut, hazelnut, and other forms of nuts. So most of the time when people are allergic to nuts or to food, it's basically because of the mutation in that gene that is actually producing a protein that is called filaggrin that is found in the skin. Now, when an allergen comes in, what specifically happens? So if we consider an allergen such as pollen, right? An allergen such as pollen is going to come and bind on a B cell receptor, right? On that B cell receptor, upon binding, well, obviously the B cell now is going to be activated. Once the B cell is activated, then what? That B cell is going to differentiate into plasma cell. The plasma cell at that point will do its job, which is to produce antibodies and release these antibodies by secretion, right? As these antibodies are released, these antibodies are going to be recognized by IgE receptors. These IgE receptors, upon binding the antibodies, well, these IgEs are now receptors are going to cause the mast cell, because they bind on the IgE receptor on the mast cells, right? This mast cell will actually now start producing some allergic factors, and these allergic factors are going to be in vesicles, right? This vesicle that you see here. However, these allergic factors are not just going to be released right away, right? You have an activation of mast cells because of the attachment of antibodies on the IgE receptors. Eventually, if there is another exposure of the cells of the body to allergens, the same allergens, right? As this allergen attach to the antibodies that are themselves attached to mast cells, well, we are going to see a release of these chemicals that are actually allergic factors. These allergic factors are going to be histamine, heparin, and eventually other chemicals as well. So this right here is going to actually trigger a cascade of events that is going to lead to a rash, redness on the skin, runny nose, runny eyes, and so on and so forth. Now, let's take a look at the immune response during pregnancy. Well, during pregnancy, we're going to have two factors. We have the fetus that needs to survive, and you have mom that needs to stay healthy. Now, realize that during pregnancy, the body of mom is going to think of the fetus as something foreign, something that doesn't belong, while the fetus, on the other hand, is going to fight for their lives, right? So they're going to do whatever it takes to actually develop completely and come to life through the birth process, right? Now, as we consider the fetus is going to be made up of two genomes. One that is from mom, two that is from dad, right? So that fetus is going to produce a number of factors. These factors, some of them are going to be seen by mom's immune system. Some of them are not going to be seen by mom's immune system. But regardless, there is a crosstalk between mom and the fetus. Now, one is that the fetus is releasing some factors that cause mom's immune system to tolerate the presence of the fetus. On the other hand, mom is also producing some hormones that actually dampens her own immune system for the sake of reproduction. Now, that is some kind of a paradoxical reaction, but that reaction is part of the evolution, right? Remember that as living organisms, the purpose of a living organism is to leave behind offspring, right? Considering that you have, in terms of evolution, mechanisms that have evolved such that our immune system, at least when I say our immune system in the humankind, right, in a female, are going to tolerate pregnancy, but that goes with a dampening, a decrease in the capability to respond in terms of immunity, so that mom is going to actually be able to tolerate the pregnancy throughout the development and until birth. But remember, and very importantly, that there is a crosstalk between mom and the fetus. There are a number of cells that are going to be involved in controlling that response, including T cells, and specifically regulatory T cells, that are going to make sure that mom is able to tolerate the presence of the fetus, but also the fetus is able to tolerate whatever is coming from mom and that is coming in, right? Now, unfortunately, sometimes this is not always possible to navigate that tolerance without any issues. We are going to see this in some conditions. Now, the kind of condition I'm talking about, it's for instance when there is what we call a rhesus incompatibility. Now, what is rhesus incompatibility? So, in our red blood cells, we have a number of surface markers, right? We talk about blood type A, blood type B, blood type O. Blood type A has antigen A on the surface. Blood type B has antigen B on the surface. Blood type O has no A, no B. But in addition to this different antigen, you have another factor that is found on the surface of red blood cells. This factor is the rhesus factor. Now, some of us have the rhesus factor, some of us don't. Now, it can happen that mom right? A woman would be rhesus negative, and having a child with a man with rhesus positive is going to necessarily give a fertilized egg that is going to lead to a zygote that is going to be rhesus positive. All right? At that point, mom is rhesus negative, and she's going to carry a fetus that is rhesus positive. Now, what is the consequence of that? The consequence of that is that at some point during pregnancy or during the process of birth, right, the rhesus factor can actually be exchanged between the baby or the fetus and mom. And at that point, mom now coming in contact with a rhesus factor, which is at that point an antigen, right? Mom is going to produce antibodies against that rhesus factor. Now, here is the thing. During the first pregnancy, right, when a lady is carrying a rhesus positive child while the lady being rhesus negative, that child is going to come to life, be born without much complications. However, the problem occurs during a second pregnancy. Why? Because during the first pregnancy, sometime before birth or at birth, mom is going to become aware of the presence of the rhesus factor. And as mom becomes aware of the presence of the rhesus factor, well, mom will start producing a large amount of antibodies. These antibodies that stand against the rhesus factor, and you have a particular class of antibodies that are called IgG, right? The IgGs or antibodies, Gs, are able to cross the placenta barrier. These IgGs are able to leave mom's blood and go in the fetus during the second pregnancy. And as they go in the fetus, these IgGs are going to target the rhesus factors of red blood cells in the fetus. As a consequence of that, these red blood cells in the fetus are going to die. Now, based on the severity of the reaction, the fetus can actually die because there will be not enough oxygen because of the number of red blood cells that are dying, not enough oxygen to actually sustain the life of the fetus. So this can lead to a miscarriage, right? And most of the time it's going to lead to a miscarriage. Sometimes, while the antibodies, IgGs, are crossing the placenta barrier and get to target the red blood cells of the fetus, the number of cells, red blood cells that are going to die, may not be enough to actually prevent the full development of the fetus. Most of the time, though, the baby can be born underweight or underdeveloped. This is when the pregnancy is carried out to the end, right? But most of the time, the second pregnancy is not going to be carried up to the end because of the severity of the attack of the antibodies on the red blood cells of the fetus. So now, today, things have changed tremendously. Now, because of all the knowledge that we have about how to deal with this kind of situation, well, ladies are actually injected some antibodies that are called Rho gamma, right? These Rho gamma that are injected are going to actually prevent mom from forming antibodies against the rhesus factor. In other words, we are cheating here. We are basically saying, well, we are going to prevent mom from ever being aware of the fact that she ever carried a fetus with rhesus positive factor on the surface of the cell. By doing what? By simply injecting this raw gamma during about 72 hours, no more than 72 hours after giving birth the first time. And after giving birth, she will keep receiving this raw gamma. When she becomes pregnant again, she will also be receiving raw gammas so that her body will never have the chance to herself develop antibodies against the rhesus factor. So this is how this problem is solved. All right, so now let's dive into how we can alter immunity. There are many approaches that we can use this day to alter immunity. This includes, for instance, vaccines. Vaccines are typically used to teach our body foreign particles. So purposefully, we are bringing foreign particles that we hope are not going to be harmful enough to make us sick, but good enough to stimulate our immune system to develop a response, so that when we get really in presence of the pathogenic factor, it can be a bacterium, it can be a virus, it can be a parasite, we already have the immunity that is needed to actually not get sick, right? So we have the vaccines that we use to actually alter our immunity. Also, we use cytokines and modified antibodies as drugs to fight off infections, including, for instance, we saw a lot of this, the injection of antibodies to fight COVID-19. Then we have an alteration of our immunity when we receive transplant of organs or transplant of tissues, right? So when we receive transplant of organ, we have to care about the compatibility. Now, even though the organ will be compatible, we still have to take immunosuppressive drugs, because most of the time that compatibility can only be so close, right? You will always have a level of difference, and that level of difference, even if it's a very small level of difference, it's still going to trigger some level of response. That's why we have to take immunosuppressive drugs whenever we receive a transplant tissue or transplant organ. Now, vaccination, it's a process that has been among us for a long time. This vaccination is typically done with altered pathogens or inactivated pathogens. It can be pathogen particles, right? It can be a full pathogen that is simply made not as virulent as the form that is causing us to be sick, right? So we have attenuated vaccines, we have inactivated vaccines, not inactivated vaccines, but inactivated toxins. There we go, inactivated toxins. In either case, this is going to lead to a stimulation of our immune system so that our B cells are going to produce antibodies in such a way that at the end of the day, we have a memory. when we have that memory, when we actually get infected by an infectious agent that is similar to the vaccine that we received, we are actually going to respond as if we had already encountered that pathogenic agent before, right? So the antigens that are chosen are typically going to be harmless, right? But what we hope is that the initial exposure through vaccination is going to actually cause a very good reaction to an ideal reaction when we actually get exposed to the infectious particle. So we saw a vaccination, for instance, a long time ago, 11th century in China. And then we had some people who are well known, such as Edward Jenner, right, who actually noticed that people who were working with cows actually did not systematically get infected with smallpox. And actually they came with the idea that, Edward Jenner came with the idea that, well, if we can inoculate people with cowpox, maybe that can actually prevent them from getting smallpox. And guess what? That's what happened. Now, obviously, at Jenner's time, they could inoculate people with pus from cow, but this time around, in this modern days, this is not acceptable, right? It's not a practice that can be in any shape and form acceptable. All right, so vaccination has been very effective. Nevertheless, many people are standing against vaccination. And the reason why many people are standing against vaccination is because we've lost awareness of what the condition of our health would be if there had never been vaccination at the first place, right? So a lot of people think that, well, vaccination is more of a problem than it is solving our problem. Well, we know today that because people are unfamiliar with the situation, including even in the health industry, right? The healthcare people, including physicians, some, for instance, are not aware anymore that smallpox is actually a reality, right? And they tend to say, well, Phil, you can immunize your child or not immunize your child. That's okay with me. But guess what? It is not okay. Now I'm talking from the perspective of a microbiologist, right? Because we do know that wherever the rate of immunization is not high, we are seeing outbreaks of smallpox, believe it or not. We see outbreaks of measles among the communities where there is resistance around immunization. All right? So, because of our success, we are now being victim because, well, we've not seen cases of measles, smallpox for a long time. Then, therefore, we say, okay, well, I guess this is not a real issue. Yes, it is. It is. So anyway, so here we have on this photograph, this lady who is actually having a bad reaction, smallpox. Okay, and you can see how she's completely disfigured. All right. So what we hope with immunization is to reach herd immunity. But what do we call herd immunity? Now, we have two scenarios here. we have a scenario where we don't have enough people in the population who are vaccinated, and here we have enough people in the population who are vaccinated. So now, healthy non-vaccinated people are here in blue. We have healthy vaccinated people who are in yellow here, and non-vaccinated sick individuals who are in red. Now, in a population where only some individuals are vaccinated, what we see is that at one point in time, when one person here is sick, that person is actually going to pass the disease to people around them that are not immunized, so that the people who are immunized are still going to be safe, right? However, everyone else that was not immunized is going to be sick, right? So that we have a large population of people now over time that is sick, and now we're going to have hotspots of the disease that are going to move from the center right here to the peripheries right here, right? And this is going to spread more and more. Now, if we look at a population that is heavily vaccinated, well, what happens is that with one person who is sick and only a few people here who are healthy but not vaccinated, guess what? With this person who is sick, if the disease goes to this person, well, this person being immunized is going to be able to confine the infection, same as going to this person or going to this person, right? As consequence of that, the infection is going to be confined, but notice that only one person really get to be sick, one additional person get to be sick. So this is what we call herd immunity. So the more people are vaccinated, the highest the chance to control an infection in the population. But the smallest number of people are vaccinated, the smallest the chance of limiting the infection in a population. So we want herd immunity. Now, there are many ways to get people immunized. We know that traditionally we do immunization by injection, right? That is the most common way to immunize people. But nowadays, we also have immunization by just introducing the vaccine on mucous membranes, such as into the nose, right? We have by eating, right? But the edible vaccines, these are very uncommon and they are still experimental, right? So in the most common, beside injection, obviously that is a traditional way, which is actually still the most common way of administering vaccines. We have nasal sprays. We have genetically modified fruits and vegetables, right? So these are ways of immunization that are new but not very common, all right? So here, a foreign antigen is going to stimulate a phagocytic cell, right? And basically, at that point, from a phagocytic cell, we're going to go to T cells that are going to now trigger a whole cascade of events. We have T cells that are going to stimulate B cells. And at the end of the day, you're going to have the production of immunoglobulin class A, right? We can have some coats to the small intestine that are going to protect against foodborne pathogens. These are some other ways of providing protection. And then you have a protection that we can provide by producing antibodies. For instance, we talked about the Rho gamma, right? Remember the Rho gammas? Okay, so how do we produce antibodies that are specifically directed against a particular antigen? And these antibodies are going to be injected to a person. So typically, we're going to have antigens, right? These antigens are going to be injected to an animal model, typically a mouse or a rabbit. So as these antigens are injected to a mouse or a rabbit or maybe a rat, right? We are going to have B cells that are going to be produced against these antigens. Now, so that's one. Two, we are going to have, on the other hand, a cancer line of cells. This cancer line of cells have this advantage that this cancer line of cells can multiply over and over and over, right? So you see the advantage, these cells are immortal. Now, what are we going to do from there? So from there, we are actually going to force these B cells, right, to merge with these cancer cells, okay, to produce some hybrid cells that we call hybridomas. Hybridomas, these are hybrid cells. These cells, because they are hybrid and because they are hybrid from cancer cells, these cells will be able to multiply to a level that is going to be beyond what these B cells could multiply. Remember that because of the telomeres, our cells, when we culture them, they can only multiply so many times. So there is a limit as to how many times they can divide, right? But the case of hybridomas is going to give them an advantage to be able to multiply for many cycles without dying. So that's one. Two, we can preserve the cells for a long time and bring them back and culture them. As a consequence of that, right, we can actually isolate different hybridomas, right? We're going to isolate, for instance, a hybridoma against antigen that we're going to call antigen A here, so the red one. Then we're going to isolate hybridomas that produce antibodies again against antigen B, another one that produce antibodies against antigen C. At the end of the day, we're going to have different clones producing different antibodies. Antibodies anti-A, antibodies anti-B, and antibodies anti-C. And each group of antibody represents a monoclonal antibody. Okay? Monoclonal antibody, meaning one single clone of antibody, so only one type of antibody is produced by that hybridoma. All right? And these antibodies now can be used to treat conditions including cancers and other conditions, obviously, right? So the use of monoclonal antibodies is going to be used for research purposes, but also for diagnosis purposes, right? Some applications are, for instance, for a lady to know if she's pregnant or not, right? So we have these days monoclonal antibodies that are used to recognize a hormone that is called HCG, human chorionic gonadotropin, right? Human chorionic gonadotropin. This is a hormone that is produced in early stage of pregnancy. And when the HCG is present in the urine in presence of the monoclonal antibody that is going to produce a color to show that, oh, the lady is pregnant, right? We have the Herceptin, right? This is a drug, actually, an antibody that is used as a drug, right? Its job is going to be to block the receptors on the breast cancer cells, which actually is going to prevent potentially breast cancer cells from multiplying, right? So to these days, there are about 30 different monoclonal antibodies that are used actually to treat human conditions of all kind, right? We have the cytokine boost and cellular immunity. Cytokines, yes, are used to treat a number of conditions. Some examples of cytokines are going to be interferons. Interferons are used in the treatment of cancer and multiple sclerosis. Interleukins 2, used in preventing or fighting kidney cancer, right? CSF, colony stimulating factors, right? They are used to boost white blood cell levels in individual with AIDS. You know that in individual with AIDS, helper T cells are dying, right? And because helper T cells are dying, well, the individual is going to have a hard time mounting a protection, right, against foreign particles. So by injecting CSF to people with AIDS, this is going to allow them to boost the production of white blood cells, right? So other example, excess of cytokines, right? Tumor necrosis factor, TNF, are basically going to be the signs that there is a disease going on. So knowing this, it is possible to block, right, the production of cytokine and tumor necrosis factors, to actually treat the condition. Now, how about organ transplant? When it comes to organ transplant, there are so many different organs that we can transplant these days. We have the heart, we have kidney, liver, lungs, cornea, pancreas, skin, bone marrow. These are all transplants that are done on a regular basis these days and can be done rather successfully, right? However, that transplant depends a lot on the genetic background, right, of individuals. So we said earlier that you need at least a matching between 4 different types of MHC for compatibility to be established, right? So because of that, we always try to see, okay, well, what are the best chances, right, for compatibility to be there, so that we are going to classify the type of transplant based on the origin of the organ or the tissue to be transplanted. We have autograft. Autograft, it's basically when an individual is donating tissue for its own sake, right? So I'm giving skin to myself, right? You are giving skin to yourself, right? So from location A to location B on the same body. That would be autograft. Then we have isograft. Isograft, it's basically obtaining a tissue or an organ from an identical twin. Now, you know that identical twins, right, except for the environment, in terms of genetics, they are completely identical, right? the environment is going to be different. We already mentioned that. So we're going to have allografts, and in allografts, we're going to have a member of one species that is going to give its organ or tissue to a member of the same species. However, in the case of xenograft, it's across species, right? For instance, from pig to human, right? From cow to human, right? From sheep to human, okay? So here we have an autograft, right? We have an allograft here from a human to a human. That would be an allograph, right? An isograph. So the two individuals are genetically identical, right? So one is giving to another, okay? Autograft, so same, right? And xenograph, so of a different species to another species, right? Now, tissue rejection can occur. When that rejection occurs, it can be progressive or it can be acute, right? The hyperacute rejection is very severe, right? And actually, if it's in the case, for instance, of kidney or heart, that can lead to death, actually. So you're going to have that the individual who is receiving the graft will have to take immunosuppressive drugs. And this only is going to be viable if compatibility is established at the first place, right? If compatibility is established at the first place. Then we have the case of, bone marrow transplant. In the case of bone marrow transplant, things are kind of like not very simple, right? Because in the case of bone marrow transplant, notice that you are actually bringing bone marrow. And in the case of red bone marrow, actually, you are actually bringing stem cells. And these stem cells are actually white blood cells. So, they can become white blood cells. And these white blood cells can actually recognize the recipient as foreign tissue, right? And as this cells from the bone marrow turn into white blood cells, and now they are targeting the recipient as foreign, that becomes a major issue, right? Becomes a major issue. And then, finally, but not least, we are going to use genomes for a number of things. We're going to use genome to fight infections. We're going to use genomes to actually track the spread of an infection and be able to fight off this infection. We are going to be able to use genomes to actually design vaccines. All right? So, in the case of infection, we know that a foreign particle brings its own genome, and the host is having also his own genome, right? A knowledge of the genome of the host is going to give us a tremendous amount of information as to what the organism has to fight off the infection, right? And knowing the genome of the infectious particle is also going to be very important, because we can actually find, based on the genome, potential targets, right? And based on these potential targets, we can be like, can we actually design antibodies against these factors that are found in the genome of the foreign particle, right? So these are going to be information that are very important and that allow us to go beyond just growing a bacterium, a pathogen, right? Just growing a virus and characterizing that virus from a microbiological perspective, right? So microbiology is great, but it is limited. Genomics is much better because we can actually dive into the genome and actually predict with some algorithms, right, what genes could encode for, right, and eventually do some kind of reverse engineering, if you will. So, and talking about reverse engineering, we have reverse vaccinology. In a reverse vaccinology, we are going to identify genes in a foreign particle that actually could encode for some proteins that can be good vaccine candidates, right? We've designed a bunch of vaccines using that approach, right? I'm not going to develop the details, but some of the work that have been done is to, for instance, produce a vaccine against the meningococcus vaccine, okay, what we call the meningococcal vaccine, right? And also another vaccine is the SARS vaccine, right? That were produced based on the genome of the vaccine. And then finally, but not least, using genome sequencing, we can track outbreaks, right? Infection outbreaks. Okay, so typically we are going to isolate a bunch of microorganisms in a given geographical area, and we are going to do what we call metagenomic analysis. And when we do that metagenomic analysis, we are going to compare the genomes that we obtain with a database, right? And as we compare the genomes that we obtain with the database, we are going to be able to see that, well, out of all this genomes analyzed, there are some where we can actually pinpoint some particular changes, the single nucleotide polymorphism, right? And the patterns of single nucleotide polymorphism, SNPs, among organisms in a not a population, I want to say a community, right? We are going to be able to trace actually how this organism was spread around, right? From one area to another area. This is, for instance, how it was possible to show that there was an outbreak of pneumonia at an NIH clinical center, and that outbreak was caused by Klebsiella pneumonia, okay? Another outbreak was also traced back to Escherichia coli in Germany, and that outbreak was caused by Shigella toxigenic Escherichia coli. In other words, this is an E. coli that has obtained features from Shigella flexneri, and those features are specifically a toxin that is found in Shigella flexneri. Now, technically, E. coli is not a harmful organism. However, E. coli can acquire some genetic information from the environment and becoming actually harmful at that point, right? This was the case in this particular situation. So this would conclude this chapter. Thank you for your attention. Bye now.